Hello! Today I'm going to tell you about a study that we did recently and that is part of my doctorate. It is called Tracking a Microscopic Invader in South America, Ceratium furcoides. I am Victoria Catatis, a PhD student from Santa Fe, Argentina. I am doing this research with my director, Dr. Melina de Berselli, my co-director, Dr. Claudia Piccini from Uruguay, and also with Dr. Paula Huber and Dr. Sebastián Metz. My colleagues from the National Institute of Limnology and the Clemente Stablet Institute of Biological Research also accompany me. In this presentation, we are going to focus on dinoflagellates, belonging to the clade of the alveolates. In particular, this study is based on Ceratium furcoides, an invasive dinoflagellate that is currently expanding in South American ecosystems. The first bibliographic record of Ceratium furcoides in South America was in Colombia around 2001. Then it began to spread to the south of the continent and radially. In 2014, the first record of this species was formally published in Argentina. The strongest hypothesis is that Ceratium furcoides came from Brazil, although this had not yet been proven. But which factor helped Ceratium furcoides to establish itself as an invader? This species presents a series of characteristics that help its dispersal: mixotrophy, the generation of resistant seeds, its size and shape in relation to the rest of phytoplankton. In addition, it can be found in high biomass given its ability to produce blooms. What contribution did we make with this work? We performed the first study of Ceratium furcoides in South America, combining morphological, molecular and phylogenetic information of natural populations from the Paraná fluvial system. This system is the second largest river in South America, formed by a main channel and a flat plain fringing its right bank, which is hydrologically connected by pulses of flood and drought. We collect samples at the main channel of Paraná River and floodplain water bodies. Our methodological path was divided into three stages. First, we carry out the morphological identification by taking measurements of total length, width, and medium biovolume of a total of 25 individuals per sample. Secondly, we did a phylogenetic reconstruction of Ceratium. To accomplish this, we isolated cells from a floodplain stream manually under microscope to conform a pool of 15 to 30 individuals. Then we did genomic DNA extraction. The before hypervariable region of the 18S ribosomal RNA gene was amplified by PCR. After agar gel electrophoresis, amplicons were cut from the gel, purified and sequenced with Sanger. Finally, we performed the detection of Ceratium furcoides by high-throughput sequencing, specifically by Illumina MySeq, in 6 environments of the Paraná River system. We defined operational taxonomic units as applicant sequence variants with data2. As a result, 3 ASVs were identified as Ceratium. When doing the morphological identification, we saw that the cells were fusiform and dorsiventrally flattened, with very different measurements between the stream and the lake, the latter being larger. We were able to morphologically identify Ceratium furcoides by its conical epitheca with a single horn and its short 4 triangular plate. This distinguishes from Ceratium hirundinella, a species that is also invasive in South America but is expanded in the opposite direction, from the south to the north of the continent. Finally, the hypovalve had 2 or 3 horns that varied widely in length. We obtained a 499 base pair length 18S ribosomal RNA partial sequence, which was uploaded to GenBank and became the first South American sequence on this platform. Plus analysis showed that this sequence shares 100% identity with the 2 available sequences from Asia of Ceratium furcoides in the database. We performed the phylogenetic reconstruction for Ceratium using the Sanger sequence obtained, marked in bold, and the available sequences of the 18S ribosomal RNA gene of the genera Ceratium, Tripos, and Protoceratium as an outgroup. The resulting phylogenetic tree showed 2 highly supported clades, one of them belonging to marine species and the other to freshwater species. Within the freshwater clade, a first node defined an European clade containing Ceratium hirundinella, then a second node gave rise to the clade containing American and Asian Ceratium sequences with 2 distinguished groups. one including uncultured Ceratium-like sequences from North America, and the other harboring the Paraná sequence obtained here together with ancient Ceratium furcoides, which formed a highly supported monophyletic clade, 100% bootstrap value. Accordingly, Ceratium furcoides of Paraná floodplain was identical to Asian sequences and seemed to be more related to its North American counterparts than to the European ones. Hence, tree topology suggests a hierarchic pattern of Ceratium species with a closer origin for Asian and South American populations. To infer their phylogenetic position, we incorporated by parsimony the sequences obtained by Illumina ISEP into the phylogenetic tree obtained above. All the sequences clustered into the Ceratium furcoides clade, corroborating the taxonomic affiliation. When we compare them with the Sanger sequences from a floodplain stream, we discovered that sequence identity was higher between organisms of floodplain water bodies in comparison with the main channel. This led us to ask ourselves, Ceratium furcoides has different environmental preferences. Are there ecotypes of this species according to whether it lives in the main channel or in floodplains environments? These are questions that we continue studying. To sum up, records of Ceratium furcoides 18S ribosomal gene are shown in the map with different colors: pink for Sanger sequencing and red for Illumina ISEP. Gray squares indicate those environments sampled where no Ceratium furcoides 18 ribosomal RNA gene records were found. The obtained results highlight the relevance of combining classical morphological detection and characterization methods with next-generation sequencing approaches, which, thanks to its depth, allows the recovery of low-frequency genetic variants and alert a potential invasion in its early stages. In conclusion, we report for the first time a phylogenetic analysis and sequence in the database of Ceratium furcoides from South America. Further ecological and genomic studies combining the sequencing of other genes would allow us to understand the processes accounting for the invasion success of Ceratium and tracing its dispersal route. This work is part of my doctoral fellowship, which is based on studying the phylogeography, ecology, and the processes involved in the dispersion of Ceratium furcoides in South America. For this reason, we thank those who want to participate collecting and sending samples or sequences of Ceratium species from different parts of the world. This will help us to analyze their phylogeographic patterns. Thanks for watching.